All right, in this segment from chapter 25, we're going to talk about genes and how changes in the gene sequences can result in changes in gene regulation. All of this can result in different structures being generated in organisms and creating different morphologies. As you can see, this figure right here just shows protein sequences. You can see the differences between many different organisms along here. This here is called an alignment. An alignment is used to compare DNA sequences. We'll talk. Right, so major changes in the forms of bodies and organisms were the result of changes in DNA sequences and ultimately also the regulation of genes that are involved in development. Studying these genetic mechanisms allows us to get some more clues into evolutionary change. Now, genes, going back to development, genes that are involved in the development of the organism can control a number of things. It can control the rate of development, the timing of changes that occur during development, and the spatial pattern of changes, so where these structures are going to develop. Heterochrony is the term that's used for evolutionary change, where there's a change in the rate or timing of developmental effects. Now, there's a number of different categories that this can happen and are described by different terms. So, progenesis, right? So, progenesis is where the development of the organism stops early. Neoteny is where development is slowed. And post-displacement is where the development of the organism begins late. Now, heterochrony can have some major effects on the body shape and structures of appendages in the body. Here's an example. So here, comparing humans with chimpanzees, we are sister species with the chimps. We do have a common ancestor as chimps, but we do have different growth rates, and this affects the shape of our skull once we become adults. So, look, if you look at the chimpanzee fetus and the human fetus, the skulls are very similar. The bones within the skull are essentially the same. Now, over the course of development, the chimpanzee's skull case, the brain case, becomes a lot smaller than that of the human. You can see there's a different size of the bones as well. However, this is due to a change in the rate of development. So basically, chimpanzees are a lot faster in terms of development relative to humans. So there was a slowing of development in humans. Heterochrony can alter the timing of reproductive versus non-reproductive organs. Chaetomorphosis is basically what's described as the rate of reproductive development is faster than the somatic cells. So remember, we've got the reproductive cells versus the somatic cells. So organisms that exhibit chaetomorphosis are going to be sexually mature before they are at the adult stage. So basically, they're still going to have juvenile structures and somatic tissues, but they're going to be sexually mature at a very early age. Evolutionary change results from alterations in genes that can control the placement and the organization of the body parts. So this is where the spatial pattern can be affected by changes in gene sequences and regulation of the genes. Homeotic genes specifically are those genes that are involved in development. These are the genes that determine where the wings and the legs will develop on a bird, or how the flower parts are going to be arranged with the final adult stage. Hox genes specifically provide positional information in animals specifically. So Hox genes are only in animals. Hox genes are an example of a homeotic gene. It's a set of homeotic genes. It's just a subset that only belongs in animals. Now, Hox genes, when they're expressed in the wrong location, this will result in body parts being produced in the wrong location. When there are mutations in Hox genes, it can turn on or off certain characteristics. So I'm going to give an example. Here, this is the Hox gene mutations are basically the suspected mechanism for the evolution of insects, right? So early crustaceans had a lot more segments with more appendages attached to them, while over the course of time, as insects arose, they arose with less segments and less body appendages. So it's suspected that changes in the UBX gene specifically over time resulted in the number of segments and appendages decreasing in that lineage. Now, changes in the regulation of genes can have an effect on the structures of an organism. So here's an example. We have the three-spined sticklebacks and the lakes. They have fewer ventral spines than the ones that live in the marine, right? So these are the ventral spines of a marine stickleback. The lake sticklebacks do not have the ventral spines or have less ventral spines. To be a change in the expression of the PITX1 gene. Now, the sequence has not changed of that gene, but its regulation is different in the two groups of fish. So here we have the marine stickleback, right, the embryo. And basically the PITX1 gene, or the result of that, or the product, which is the protein, has been labeled, and you can see that the gene expression is found in the mouth area for both of them. So this is the marine and this is the lake. However, in the marine stickleback, you can see PIT1X gene being expressed in the ventral region where those spines would be located or they're being developed. It is not expressed in the ventral region in the lake stickleback. So you can see a difference in the regulation, or it's an expression of genes can affect characteristics or body forms of different organisms. Now, it must be noted that new structures are not going to be evolving in anticipation of future use. This is the thought behind intelligent design, right? So those that believe in intelligent design believe that the use of something is known and basically reading the future, right? So it's going to be known. So we know that the use of this will become necessary. So then that structure will develop. That's not really how, there's no evidence for this. Now, evolution is basically just like tinkering, right? So it's just a process where mutations occur and new forms may arise, or there might be some modification of a form that already exists. This is the modification of a form that already exists is called an exaptation, right? So exaptations are structures that evolve for one purpose, okay, or one function or one context, but then evolve for a different function. Now, natural selection basically just improves upon the structure that is already there. So the genetics or the component must be there. The gene sequences which result in this structure must already be present for natural selection to select for or against it. Now, one example here we have in terms of eyes, right? So there's a cool video which I have posted in the cool videos section of this chapter resources about the evolution of the eye. Now, many interesting novel biological structures evolve by exaptation, and the eye is one of them. So, the complex eyes, which we have and other more advanced organisms have more complex eyes, they evolved from simple photosensitive cells. And this occurred independently along different lineages. So, organisms that exist today still have these simple eyes, right? So, we have the limpets, they have just basically simple a collection of photosensitive cells which allow them to detect light. However, over the course of time, more complex eyes have developed, and you should watch that video, which describes the evolution of the eye. Well, this concludes the effects of changes in gene sequences and gene regulation. As you obviously listen to, that you can see that changes in gene sequences can result in the change in developmental processes in terms of timing, the spatial development, and regulation of these genes can also affect the same. All right, thank you for listening.